Hey team, Dr. Jack Gordy, and in these few videos we've covered how cells and tissues respond to stresses. Now I want to go through a very specific example, and that will allow us to dive a little bit deeper into how the body actually figures out how to respond to the stress. How does it figure out there is a stress, and then how does it respond to the stress? Now I'm going to focus on one specific stressor, and that is hypoxia. Hypo meaning low, and oxia meaning oxygen. So low oxygen. We can see that this person's lips have gone blue. That is a sign of systemic hypoxia, and it's a big red flag. If you see this in any medical setting, we're in big trouble. And actually, I have seen this in my child. He underwent an anaphylactic shock, which is an allergic reaction. His throat's swelling up, so he can't breathe that very well. And so he's undergoing hypoxia. But don't worry, we gave him the EpiPen that opened up his airways and he could breathe and he was fine. He immediately started crying. It was terrifying, though. Absolutely terrifying. Little side note, your blood doesn't actually go blue. It goes dark red when it's not oxygenated. However, when light comes in through our skin, hits the dark red, and then bounces back out again, it's that filtration process is actually what's promoting the blue color. So it's actually our tissues filtering out a lot of the red light and giving you that blue signal. So if you see blue on the lips, it's not because the blood is blue, it's just darker. And the white light is bouncing out as a blue light because of that dark. Okay, so let's jump into hypoxia. Let's have a look. Right, now I'm actually going to zoom right down to the molecules and cells. Here we go. So here we have a cell, there's the membrane, there's the cytosol, here's the nucleus, and here's some DNA. Now in most cells, in many cells, we get what's called a constitutive production of a protein called HIF-1. Now, constitutive production means it's just always on. So we normally get this little constant trickle of the production of this protein called HIF-1. Now, HIF-1 stands for hypoxia-inducible factor 1. Hypoxia-inducible factor 1. HIF-1. Okay, so that just goes through transcription and translation, and we end up with a constant expression of this HIF-1 protein. However, in the presence of oxygen, here it comes. Here we go. We've got oxygen. Oxygen can actually react with HIF-1, and it will create, one of the oxygens will come off and it will create a hydroxyl group attached to the HIF-1. So that's an OH group just attaching to the HIF-1, and that came from oxygen itself. So when there is oxygen, one of those atoms will come off the molecule and form a hydroxyl group on HIF-1. Now, this hydroxyl group actually attracts a protein complex. Now, this protein complex, what this is going to do is it's going to start to attach these little molecular tags called ubiquitin. Now, ubiquitin is named because it's ubiquitous. It's everywhere. Every cell is full of ubiquitin. And what ubiquitin does is it slowly tags all the proteins in the cytosol. It slowly binds and tags to them. However, this process can be sped up through proteins which increase the ubiquitin rate, and it can be slowed down with proteins that remove ubiquitins off protein. So this green protein here that's attached to HIF-1 is bringing in ubiquitin, and it's tacking it onto HIF-1. So now it's got all these molecular tags, a long chain of these ubiquitin molecular tags have been attached to HIF-1 alpha. Now what does ubiquitin do? Well, what it does is it pulls protein into a protein machine called the proteasome. Now, the proteasome is a protein machine. It's quite large. That sucks proteins in and it chops them up into a million bits. So the ubiquitin chain can drag proteins into the proteasome and chop them up into a million bits. So in the presence of oxygen, we get the hydroxyl group. We get another protein that comes and joins. It's attracted to that hydroxyl group. It will attach ubiquitins to it. The ubiquitins will then drag it into the proteasome and mince it up into bits. So this is what's happening in almost every one of your cells in your body when you're breathing well and all your cells have lots of oxygen. This is what's happening. Now, what happens when we have a low oxygen environment? So we still get that expression of HIF-1 because remember, it's constantly just dripped into the cytosol. It's constantly being translated and transcribed. But this time we don't have oxygen. So we don't get the hydroxyl group forming, we don't get the protein complex, we don't get the ubiquitination, and we don't get the getting sucked into the proteasome and minced out the back. So what we actually end up with is a buildup of HIF-1 protein. Because there's no oxygen to lead to its breakdown, we end up with a buildup of HIF-1. Now, HIF-1 can then go into the nucleus, and it's actually a transcription regulator. So what that means is it helps to mount the RNA polymerase and turn genes on. There's specific sequences of DNA that tell HIF-1, I want you to bind there, and I want you to bring an RNA polymerase on, and let's start translating this gene. Now, HIF-1 can actually bind to lots of genes to regulate lots of proteins and flipping lots of genes on. So it can turn a lot of different genes on. It's basically a molecular switch to turn a whole bunch of genes on. And what genes it turns on depends on what tissue we're in. So it'll start to transcribe and translate a whole new set of genes in response to the low oxygen environment. But here's the question, what genes do they turn on? Well, we need to start talking about what cell type we're talking about to get an accurate breakdown about what genes are being turned on, because it does depend on cell type. So let's have a look at muscle cells, because we just touched on them with hypertrophy and atrophy. So let's have a look at muscle cells. This is a zoomed up version of a muscle cell. We've got those muscle fibers, those long proteins running the length of it. We've got ribosomes. Ribosomes are important to produce those long fibers and to produce proteins. So a growing and active muscle cell will have ribosomes in it. And we've got HIF-1 bound to the DNA. So this muscle cell must be in a low oxygen setting because the HIF-1 has survived, it hasn't been digested, and it's gone into the DNA, and now it's turning on some genes. So what genes is it going to turn on? Well, actually, it's going to turn on a whole bunch of genes that start a cellular process. They're either involved in it or they signal it as well. So these proteins are going to start a cellular process called autophagy. Now, auto means self, and phagy means eating. So HIF-1, because it's hypoxia, has started a process of the cell to self-eat. It's going to eat itself, okay? Autophagy, eating itself. So let's have a look at how that works. Well, what happens is it forms an autophagosome. So this is a stretch of endoplasmic reticulum. So it's actually got two membranes. It's like a tube. Imagine a sausage. And then you bend it around and it will connect at the end. And it will encapsulate a lot of what's in the cell in the process of bending around and connecting to itself. Hopefully I've got some sick animations for you guys. Here we go. So you can see, yeah, the autophagosome is going to stretch around and eventually it's going to maybe wrap up some muscle fibers and wrap up that ribosome. And eventually it's going to wrap it up until it forms a bubble. And it's actually a double layered bubble because it was, imagine two membranes like that, and then you wrap them all around like a sausage, you're going to end up with an inner and an outer sphere. Now that's an autophagosome. And then what happens is digestive enzymes and acids, H+ ions, it's going to get acidic. It'll get acidic, and digestive enzymes are going to go into that autophagosome, and it's going to chop up the ribosomes, chop up the mitochondria, chop up the muscle fibers there. And essentially it's just going to eat itself, and then it will disappear. And then what will happen is as it eats itself, the cell size is going to shrink. And so what have we done? We have done atrophy, guys. Here's some nice full muscle cells, and here's some atrophy there, and it's all been activated by HIF-1 alpha. So HIF-1 alpha, the low hypoxia environment, has induced atrophy in these muscles. But get this, HIF-1 can do even more than that. Isn't this crazy? HIF-1 can do even more. It's also going to produce certain proteins that are signaling molecules. And these proteins are going to go out into the extracellular space, and they're going to signal the growth of more blood vessels. So they're going to promote vascularization of that muscle. That's awesome, right? It's the muscle said, hey, I'm low in oxygen. Maybe I need more blood vessels. That's pretty cool. And so here we can see the sort of body transformation, long distance endurance training, thin, small muscles. These muscles will be highly vascularized, right? So these are small, efficient muscles, highly vascularized. The person doesn't weigh a lot. You don't want to be dragging 120 kilograms around a full marathon. That's not a good idea. So you've got small, efficient muscles that are highly vascularized. A sprinter who's sprinting in 10 seconds, 100 meters, isn't going to get that hypoxia feeling. It's only 10 seconds. Your body has those reserves in oxygen. It's going to be fine. So a sprinter is much more likely to get those much large muscles that can generate much stronger forces because there's more muscle fibers. And so you can see by long distance running, you create a hypoxic environment and you promote that kind of body type of atrophied muscles that are highly vascularized. Now, that's HIF-1 being good. HIF-1 can be bad. So let's just talk a bit about cancer. Now, you're going to get much deeper dives on cancer in other videos, but cancer can largely be seen as massive cell division, unregulated cell division. So cells divide, 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 divide. Now, they don't do it properly. If your body was dividing like you're growing, it would do it properly and it would grow the blood vessels and it would divide and there would be the right factors to grow the blood vessels. But when cancer does it, it divides, 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 and doesn't properly vascularize itself, doesn't form the right shapes. You know, it's just unregulated growth. And so what happens is in a tumor that's divided and divided and divided, it doesn't have blood vessels getting into the tumor, those divided cancer cells. And so you actually end up with a hypoxic region. And sometimes it's so hypoxic that the cells die and they form like a goo inside the tumor, which is really not great. It's not great because that causes mutations, promotes more cancer. Anyway, the hypoxic region though is hypoxic, low oxygen, right? And so HIF-1 is going to start to get switched on. It's going to start to switch those genes on. And it's going to produce those growth factors. Here it's actually labeled it, VEGF, but don't worry about it. That's just one of the major growth factors that promotes the vascularization of that cancer. So HIF-1 is actually allowing the cancer to get more energy, more nutrients, more blood supply to grow and grow that tumor even bigger and properly vascularize itself. And because of this, HIF-1 is actually becoming, there's research going into it being an adjunct cancer therapy. It's never going to cure cancer by itself, inhibiting HIF-1, because you don't want to vascularize those cancers. So it's never going to become a treatment in itself, but it might help. So you might be giving multiple different kinds of drugs, and one of them is to prevent the vascularization of those tumors by inhibiting HIF-1. Very clever, very interesting, but it shows you the double-edged sword of the adaptive processes. So HIF-1 helps vascularize your muscles, also helps vascularize cancer. Not great. All right, let's look at another role of HIF-1, but this time in different organs. So let's imagine we're systemically low in oxygen. Maybe we're in the last few kilometers of a marathon. Maybe we're running in a mountain range where there's low air pressure, and so there's low oxygen getting into our blood cells. So maybe let's just imagine we're in some sort of low oxygen environment, for example, long distance running. What's going to happen is our kidney, mostly our liver a little bit, HIF-1 is going to turn on in those organs, and it's going to promote the secretion of this signaling molecule called erythropoietin. Erythropoietin. And this is a signaling molecule that's now going to get turned on and released. This is a signaling molecule that will go out into the bloodstream and it will go round to our bone marrow. And in our bone marrow is where red blood cells are produced and white blood cells. And it's going to tell our bone marrow to pump out more red blood cells. So erythropoietin, also known as EPO, is a signaling molecule regulated by HIF-1 that's going to signal we need more red blood cells. We're not getting enough oxygen to our kidney and to our liver. Therefore, we need more red blood cells to sort of get that oxygen from our lungs down into our liver and down into our kidneys. So the EPO is going to signal that. So now we're going to end up with loads and loads of red blood cells. And what can happen now is a negative feedback, homeostasis. So we might increase our oxygen carrying capacity, which inhibits HIF-1, which will inhibit the expression of EPO, which will inhibit the red blood cell proliferation. So that way we can find a nice balance depending on our oxygen environment and how often we're stressing it. But check this out. This is crazy. So HIF-1 was causing atrophy in your muscles, but it's causing hyperplasia. The increase in cell number within a system, organ, or tissue is causing hyperplasia of your red blood cells. Isn't that amazing? So HIF-1, it shows you how it can do different things in different contexts to sort of perfectly adapt our body to a low oxygen environment. It's going to shrink our muscles, which use up a lot of oxygen, increase vascularization to make sure the low oxygen organs are getting those blood vessels, and pump out more red blood cells to carry around what little oxygen is available to make sure we're getting every bit of that oxygen out of our air, kind of thing. Now, you might have heard of EPO, particularly you might have heard of it because of this guy. Lance Armstrong, he won the Tour de France a whole bunch of times. He was taking exogenous EPO, so he was injecting it to produce more red blood cells. There's actually, if you read his biography, there's a gross passage in there where he described lying on his bed and he felt like he could feel his blood struggle to go around his body. He said he could hear it, and it was like a thick. And when that happened, he felt like his blood was going to stop because it was so thick with red blood cells. So he would get up, drink a whole bunch of Powerade, and get on his bike to vasodilate and get his blood pumping again. And he was terrified of having such thick blood because he was giving himself exogenous EPO to get those red blood cells. Pretty interesting, but isn't that cool? HIF-1, HIF-1 alpha is the cytosolic one. But anyway, HIF-1 is one of those amazing transcription regulators. It's sensing the amount of oxygen in your cells, and it's inducing the appropriate response on whether it's muscle atrophy or pumping out more red blood cells. So it's doing these fantastic responses to the stress of being hypoxia. However, it can help those tumors by revascularizing those tumors that are hypoxic too. So it's a bit of a double-edged sword. Thanks, team, for learning about how the body responds to these stresses and following me through on this one example. If you have any questions, just let me know.